Hello, my name is Sabrina Templo. Today is May 27th, 2020, and this video is for Bio 225. This video is for my results or my reflection for microscopy for microbiology. And for this week, we got to know the microscope. We also learned how to look at prepared slides, and we also learned how to make our own wet mount slides. That I found it was pretty cool to label all the parts of the different microscope and get to know the microscope itself before we got to do anything else. I found it challenging to figure out how the field of view and the magnifications itself, getting to know it. It did take a little while for me, just because it was a while before I played around with a microscope. The last time I held a microscope or looked through a microscope, I was probably 17, 16, and that's been over 20 years old. So let me go through the slides. So the amoeba was the first one that I did. It was very challenging for me to see. I'm not sure if it was the size or the color. It was easier to see on the 5X and the 15X, but it took me about 2 hours to finally find it on the 40X, but it was a very pretty magenta color. The next one is the Penicillium with Conidia. And this one was my favorite slide to see and find, just because it was easy to see on all three magnifications. It looked like a very pretty blue feather, and you could see all the detail on it, and I couldn't really stop looking at it. It was very pretty, like I said. It looked very magnified. Looked like an HD on TV, and it looked very pretty. Again, the third one I did was the yeast hole mount. It was very challenging as well. I'm not sure if it was just hard to see, or if I did go with the technique of going with a lower lens and then the medium and the high power. It did help a little bit doing that technique, and I did that every single little time while looking at the slides. I did circle as well the whole specimen, so that was my technique. The next one is the bacteria spirillum, and then this one, it was very hard to see. And then this one, I really needed the oil mount, and it just brightened up the picture of what I was looking at under the microscope. So this time I was going from the low to the medium to high, and with the oil, it really just helped. The next one is the bacteria bacillus form. This one was very nice. It looked like a lot of dots, actually, like a bunch of confetti. It wasn't colorful though, and it was very nice to see the different shapes, the sizes of the different bacterias. And then the last one is the bacteria coccus form. This one kind of looked like bits and pieces of sand scattered to me that was under, and I was able to see the clearest for all the slides on the 40X, which is equivalent to 600 X magnification, because it's multiplying with the 15X. And it was quite difficult for me doing this experiment, just again because it was hard, just because I wasn't used to using the microscope itself. And I didn't know what I was looking for. It was kind of hard, and I was like really nervous, so maybe that made it difficult as well. But I'm excited to continue and practice more and how to look at slides, so hopefully I can get better at this. And that concludes my reflection video for this week. Thank you.